What keeps me going? I think it's the pressure from failing. You fail once, but if you give up, you fail
Science is for
forever.
everyone, all backgrounds, all ages, all genders, and all walks of life deserve an education.
So
Come be
Charlie,
one of
please,
us.
will you mind giving us a quick introduction? Who you are, where you're working, and just, what is your big project? What is the big question that you're currently trying to ask in the lab?
Sure. So I'm a postdoc, a new one in Dr. McGarry Houghton's lab at Fred Hutch Cancer Center. And the big question that he hired me to answer is it's actually really cool. And it's something that I guess there's a little bit of background I need to go over is like, how do we beat cancer, right? And, you know, it's really hard to balance curing cancer and, the convenience of human. Cancer is something that's always growing inside of you at all times. It doesn't take a break, but humans don't want to be in the hospital at all times. They want their treatments in convenient boluses. They want to feel side effects. So where there's a lot of considerations that have to go into medicine development that, a growing tumor doesn't care about. And so how do we get that 24/7 surveillance inside a patient? And, unless we're talking Star Trek and nanites and all that stuff that enters your blood, we have biological nanites in our body now, and those are your immune system cells. And so, Dr. Houghton here in this lab, he studies a very important subset of the immune system called neutrophils. And neutrophils are actually most important for fighting infection. But cancer actually has repurposed neutrophils into turning down the immune cells that would normally clear cancer and kind of use them as a shield and a deterrent for those immune cells to clear the cancer cells. And then they can live happily and grow, unfortunately, inside the patient. So my work right now with Dr. Hone is trying to figure out how can we turn those neutrophils off inside cancer patients, but still maintain their good effects, which is, to fight infection.
So is it a question then you would say of targeting as much as it is activity? Like how does like because it seems like there's multiple avenues here that you're going to have to modify. What are like all the different pieces then that when it goes into something like this? Because it doesn't seem straightforward. It doesn't seem like you can just modify one element of the cell to be able to have this targeting.
Yeah, that's a good question, and I definitely don't expect it to be just one thing, and it'll probably be a combination of no less than five contributing genes that make up the phenotype of how neutrophils protect cancer, and probably for the span of my whole career, I might only address two of them because
Wow.
that's the pace of science. It's very slow, but
So can you
it's slow to do it right.
I think it's really cool just like of how you have the number of like well it's five things like five potential genes. Is that based off a screen like a screen or mutagenic screen that you had done before, or is that just like a guess of like look based on the literature here's all the different things that it could have been?
Boy, I think it's definitely a hunch that I've been doing science for 15 years, and it's I guess it's a number I kind of pulled out of my nowheres, but also out of my intuition. It's not a million genes. We don't even have we don't have that much, and it can never is just as simple as one. And you're right. I totally made that number up, Lynn. I can't.
No, no,
I
no. It's
can't.
just, but it's important, right? Because some of the work that goes into this, right, is presumably there's screening techniques or literature searches, looking at other data sets. Like you are not an island, I hope, right? Like there's, you get to build on work that others have done in the past, or even do big, almost fishing expeditions to try to find candidates to pursue, so that you can know what to research. Otherwise, it's like, where do you even start, right?
Yeah, and this project that I'm doing with McGarry is actually I think it's so novel that I can't really say based on precedents where we're going to end up because it's
That's
so
that's the
novel.
best kind. That's so
If
cool.
I if I may overshare, it's a little bit off topic, but also alarmingly on topic. Is when I was a graduate student, I went to a conference in Japan, Yokohama, and it was a gastric cancer conference. This is my old work with Dr. Sandra Riome, and I'm watching these world they're the leaders of the field in gastric cancer, both on the surgeon side, for the MD side, and on the research side. And it really jarred me when they're talking about their accomplishments over the past 20 years. And all they've managed to do is shift the survival treatment needle of patients from, give them an extra 3 months to live.
Wow.
And they're clapping, they're celebrating. And I might have been, to my knowledge, the only person that I had a grumpy face on. I was like, this is not what people expect. It's not what people deserve.
Yep.
We don't pay scientists for 15 years to move the needle 3 months. And I think some real novel ideas need to come into research. None of that we call them Me Too papers. Is where and a discovery is made in one cancer, like pancreatic cancer, and we simply just take that same treatment or idea and Me Too it into another cancer type, like gastric cancer. And if it never cured pancreatic cancer, the hopes aren't high it'll cure gastric cancer. And so, this is a little bit from my personal, like dark opinions, that you don't just say out in the scientific community. But we need to do more, and we need to think bigger. I'm not happy with giving patients just 2 months to live. I want to cure cancer, and that's why I'm doing immunology work.
So you mentioned your earlier work was also in gastric cancer. So then does that mean like throughout your journey, have you always been interested in that cure for cancer, like in your research journey?
That's very funny. My research journey is wild. I started at UCLA actually studying anthrax, and
Whoa, okay.
back then anthrax was the hot stuff. Like supposedly Saddam Hussein was using it, and Bush
Yeah.
invaded a country over it. And my lab, Dr. Ken Bradley at UCLA, we had tons of funding, and then suddenly that funding dried up. I was there for 7 years, and when the fear of anthrax as a terror weapon went away, the funding for anthrax went away. Our lab shut down because the government stopped funding it, similar to many cancer labs today, as the current administration is defunding cancer research. And you know, I realized I wanted, it was jarring. All my work, all the work of me, my friends, my colleagues, it just goes up to smoke just suddenly because the money disappears. And so, one is these donations and philanthropy of the colony and donors is super important. And I determined, like I was determined to pick a career that wouldn't have this instability. And I thought it was cancer, but I'm now being proven wrong. The other disease that I think is also strongly in need of addressing is neurodegenerative disease.
Mhm.
It's one of those diseases that, like cancer, will get all of us very unjustly. It's unfair and it's terrible. And so, I flipped a coin on being cancer researcher or neurodegeneration, and here I am.
Do you feel that you made, from an intellectual perspective at least, a satisfying choice? Like, do you find it rewarding, or is it one of those things, kind of how you set it up of, I would love to be able to do these things, but that might be down the line, like the big grandiose, I can make you live longer than 3 months, right? I got you 10 years or something along those lines.
Yeah, I definitely hope, that my work comes of something, but
Mhm.
those are probably the famous last words of many scientists. Not everything we do succeeds, and that's just, we have a hypothesis, we try to see if it's true or not, and if we're wrong, that's just the way this universe was created. And so we don't always get it right. You do want to design experiments, though, so that no matter what the outcome is, it is hopefully both informative and useful. But those are kind of, I might be over speaking on scientists all over. Is those kinds of safe hypotheses are great, but they're also, I think, part of the problem as to why we move kind of slowly. You make big hypotheses and maybe you get big payouts. You make little hypotheses and maybe you get little payouts.
I like your analogy too of, if you find something and then you repeat it in the exact same cancer, right? That could get you a paper, and
Yeah.
that's our currency in science is publications, but you're not necessarily moving that needle that much more forward. So, how did you design your research question with that mindset in mind? Like, how, because it's kind of a big deal, right? You're going into lung cancer, and the immune
Yeah.
system, right? And so
Yes.
how did you design your question? And
You know, yeah.
yeah,
Yeah, no, that actually, so it's a practice that I started when I was a grad student. We want to make I wanted a thesis project that's hot, sexy, big paper, goes in the right places and makes my career. So when I went to that drawing board, and I didn't do any cancer before starting at Penn. I was in anthrax, and so coming at it with fresh eyes, actually, I would argue was the most helpful thing. So I didn't have a lot of the biases that the field had come to be afraid of. And so, one of the biggest things, especially for cancers inside the body, like skin cancers, when they're growing, you can see them. And if you can see them, you can go to your doctor and get it treated.
right.
But the problem is those tumors inside your body grow and they get so big that by the time the symptoms of those tumors show up, it's too late. And so I wanted to address that problem, which was how do you diagnose these internal tumors? And so, I would join Dr. Riom's lab and she studied gastric cancer. And so I thought, is there a cool way to do it with stool? And there is one other stool test for cancer. It's called Cologuard. It's for colorectal cancer. And I thought to myself, why aren't we doing this with gastric cancer? Everyone says gastric cancer is so hard. The only way to currently look for gastric cancer is you have to go under, you have to go to the hospital, they put a camera, they endoscope you. It's very tedious. It's actually and a huge burden on the healthcare system as a whole, but that's the only real way of looking for gastric cancer today.
Wow.
And so I thought, why don't we look for it in the stool? I mean, we're already doing it for the colon,
Mhm.
and it's just, stomach, small intestine, colon. And so it's the same testing substrate. It's stool, it's poop. So when I said that, a couple of my thesis advisors were like, "No, Charlie, that's not possible." And I'm like, "Well, why not?" And she's like, "Well, people just don't do it. It's not possible." And I thought to myself, that's not very scientific.
No.
It's yeah. So and there are definitely reasons for their fears. There's so much digestion that occurs in the small intestine that what really biomarkers can really come out of that. But I had to make the argument that I personally felt like mass spectrometry and other methods of, biomarker analysis have also evolved and come a long way since those initial biases formed. And so I was able to leverage a collaboration with Michael Gilbert. And actually, speaking of Michael Gilbert, he actually graduated from Shelley Berger's lab. She studies ants. I don't
Yeah,
know
I
if you've heard
actually
of.
I worked with Michael.
Oh my God, small world.
Yeah.
So Michael's able to, Dr. Gilbert's able to do mass spectrometry off, I think it was like picograms of protein. I mean, this is the level of sensitivity that was not present when people were too afraid to look for biomarkers in the stool for gastric cancer. And so, ultimately, I found a panel of proteins that correlate pretty well with patients with gastric cancer. And one day the hope is, okay, we can see the internal cancers. Let's get to curing them. But then you look at the cures that are available, and that was kind of paltry as well. So I kind of shifted my career focus from identification, diagnosis to trying to find a cure.
So that's really cool. Just again, that graduate student backstory, I think it really highlights that innovative mindset that you have, where it's like if someone tells you, no, this isn't going to work, you actually need a reason, right? Like, I really like the story because it's like, oh, well, stuff might break down with the pH difference in the gut. And you're just like, well, okay, but we haven't tested this in a long time and the equipment's so much better now. Why don't we just give it a shot? And then here you go, you identify a new way of screening for cancer, right? For one that didn't have that screen, that was all this inconvenience of like going in with a scope and checking. Like, that's huge.
To be fair, Sandra did tell me, she was like, look, you can do your poop thing if you want, but you got to do my blood thing first, and you can do them together. And it wasn't until I showed Sandra that preliminary data that showed her it was possible that she allowed me to move my whole thesis project into stool. So it definitely takes more than just, you got to make a little bit of pod data. But yeah.
Yeah, I think that kind of highlights of what like, y'all, this is the kind of person you want studying something that's like a disease question like this, because you want that innovative mindset. So with this like really unique mindset, Charlie, that I would argue most scientists don't have right nowadays, in part because of the funding situation, right? And I, as an alumni of Penn, I can say this as well, right? Is that there is this fear, right? Because if you lose funding, you won't necessarily be able to get funding, and then that's going to be like a long-term issue. So, with that in mind, how did you design then? You're shifting now to treatment instead of diagnosis. How did you design then that research question? Because I feel like one could argue that's even harder to start with, especially taking that innovative approach, because it's like, well, Charlie, the best we could do is 3 months, sir. So
Yeah.
how do you come up with something where you're like, if I figure this out? First of all, what do you get? Where's that if coming from, right? Like, what is the question? And then I will have a potentially large payout in terms of helping people.
Yeah, that's a great question because in immunology is not a new field. It's been done for, 50 years. And so who is some guy who studies poop to come in to say, "Oh, here's a somewhere that you guys aren't looking at. This is a novel idea, arguably a good one." And so and ultimately maybe 5 years from now it might be a terrible idea. I won't know that, but today I think it was a great idea. It's a great idea. And so how so actually it was kind of luck that I originally joined a lab at MD Anderson in Houston, and the PI had handed the project that I originally was going to be a postdoc there for to a grad student who joined the lab, and that kind of thrust me into a position where I no longer had a project, and so because of that I had to make up my own. And actually what a wonderful opportunity because I did a ton of reading, just weeks and weeks of worth of reading to a field I knew nothing about. Cancer immunology is something that I didn't know about at the time. I was not an expert in at the time. And so I thought, everyone is so busy curing cancer in mice. Just what cures cancer best the first time?
Mhm.
That I just realized, first of all, they're so prejudiced on that first time, they never think about what next. And um, because I was kind of new to the immunology field, I was looking at like, well, everyone's so focused on curing cancer the first time. As humans, we'll get cancer more than one time.
Mhm.
Even if a doctor cures your first cancer, you could get a second one. And then, the way that experiments are designed today, they assume that the mice gets the cancer just one time. And,
Mhm.
most of our work is in mice and potentially monkeys and such if the research is good and founded and solid. But, I thought to myself, maybe the cancer that a patient comes into the doctor with isn't their first cancer. It very well because we have trained cells in our body to kill cancer, T cells and natural killer cells. And so if they do their job, I thought, okay, the cancer that a patient actually comes to the hospital with is potentially the 10th cancer that forms. It's just the first one that fails to be cleared by the body's own defenses. And so I'm working on a model actually to instead of looking at mice who are getting their first round of cancer, and they say we've cured cancer in mice 1,000 times. Somewhere between that and humans getting cured cancer 0 times, there's a disconnect. And
Right.
we need to go back to the drawing board and identify what that disconnect is. And this is just one of my attempts to figure out, is it this? Like, we assume it's the mouse's first cancer, like we assume it's the human's first cancer. But what if that assumption is wrong? So that's a little bit of teaser into what I'm trying to do right now. There are definitely technical hurdles, and I'm glad Dr. Hone's very patient with the setup. But I think the question is very interesting.
It's a it also kind of feeds into the immune question too, right? Because if you've had cancer multiple times, right, the immune system may or may not have been trained or habituated towards something, right, which could be leading towards or lessening tumors on a later time point, right? So it's like there's a lot of implications just already there with the suggestion. So like it's a really interesting question. Where does one start? Do you is the idea that maybe you don't want to use it in mice, or is the idea that maybe we want to use mice and see if they have cancer multiple times? Is it a mixture? Like for you say, from the technical perspective, there are some challenges. Where do you start in actually making the research question? Like where you can actually start testing things?
Well, yeah, well, it's all about control and the ability to be able to trust what you see. So I need to develop a way, which is what I'm trying to right now, to reliably and assuredly clear cancer every time I need it gone, so that I can take a tumor naive mouse and give it cancer 2 to 10 times and watch how that immune system evolves after each exposure. And the hypothesis is that it gets weaker every time, such that by the 10th time it occurs, we've got the outbreak of tumor growth that patients present with in the clinic. And that's what I think is going to happen.
And that's again kind of harkening back to the hypothesis that you're going to be getting, this isn't the first tumor you've ever had, that the one that we see. This is like potentially the multiplied by whatever factor that you've gotten that we've found in you, and you're getting it now because some kind of defense may have failed.
Right, exactly. And neutrophils are just, you know, there is a little bit of preliminary data to suggest neutrophils and some other immune cells. But Dr. Houghton is an expert in neutrophil biology, and maybe he'll let me take maybe a little bit of neutrophil stuff and other stuff, other cell types with me. Hopefully, if I start my own lab.
So what is, I guess, beyond the fact that the lab that you work in is an expert on neutrophils, what is the maybe reason as to why looking at neutrophil science is really exciting for this potential question? Like what does the neutrophil do over other cell types in the immune system, and what might they be doing in terms of cancer, like that context?
Yeah, that's a great question. So neutrophils, number one, they're the number one most common immune cell in your whole body. In
Okay.
fact, your body makes more neutrophils per day than it does red blood cells. Just to tell you how many neutrophils the body is making is astronomical number. And so the body, and no living thing has evolved to waste this much energy if it wasn't important. And the neutrophils, one of neutrophils' primary functions is to, one, kill infectious agents, but also tell the immune system to calm down when it needs to. And like I said before, cancer has adapted that in a very, at the same time situation. Like when scientists observe tumors, there are tumor promoting neutrophils in there telling the T cells to don't kill this tumor. And so they are very likely suspect for this phenomenon because of their normal function.
So normally, even if you don't have any kind of infection, you have these cells floating around your bloodstream, right? They're just kind
Yeah.
of on patrol, and they're regulating, background immune activity. Let's just say, like you were
Mhm.
saying. So does that mean potentially the work that you're doing now, right, could possibly be even put into other disease models as well? Like, because you can hypothesize like autoimmune disorder, right, comes into play if it's this background regulation of, I'm kind of already spinning forward of like, if I've triggered these cells too many times because I've had too many tumors, where else might they be going haywire? It's not just targeting tumor cells, right? It's all of a sudden you're finding out fundamental biology of long-term stimulation of neutrophils that could have impact across many disease states. Is that me being grandiose on this, or do you think that there is something
No,
to that?
but Lynn, that's a super good point. And I'm so tunnel visioned on trying to just figure out my model in cancer that I didn't even think of that before you said it. 100%. These genes or whatever I end up discovering the phenotype to be in my model will undoubtedly also be used in other probably pathogenic situations.
That's
Yeah.
it's such a cool question that has so many implications that are already coming out. We have a question from the chat, Charlie, of
Oh,
what are the neutrophil structures like? Like, are they nucleated, unlike red blood cells? What are you dealing with when you're trying to look at this cell population? Can you look at RNA? Can you look at chromatin? Like, what are the tools at your disposal to figure out this question?
so neutrophils are very unique in two ways. One is they have this almost like a spaceship like nucleus. It's like lobular. It canonically comes in three lobes. And another name for a neutrophil is the polymorphonuclear cell. And so it looks like it's got, instead of most cells which just have one round nucleus, a neutrophil has like a three lobed thing, looks like a spaceship. And so um, it's very cool. And in fact, the lack of rigidity in the nuclear structure allows the neutrophil to get into places that other cells cannot get to and or cannot get to quickly or easily. So actually, my mad genius of a PI, Dr. Houghton, has this idea that if we can give this neutrophil like nucleus shape to other immune cells, it will give those immune cells the ability to traffic and be just as universally, available as a neutrophil is.
That's
That's
really
also
cool.
something that we are, and it's very refreshing to finally work with a scientist who is as they thinks big. Think big or fail. We're gonna think big or fail or succeed by giving someone 6 days to live. I think it's just I like it here.
It's really neat to be able to hear that connection too, right? Because if you're looking at these basic or fundamental features of the neutrophil, and you're like, okay, this might be the feature that allows them to do X, right? Like it's almost like you're saying it's not just a tunnel vision on this. It's like, okay, this is an important biological phenomena. What if we, right? I love that question. And it's not something simple for the chat, right? Like you're talking about the nuclear rigidity. So the nucleus that holds the genetic information has structure to it. There's the nuclear lamina that makes that circle, and we know that if you perturb it in disease states, accelerated aging can happen, and you could have these weird,
Criteria, yeah.
the bleb-shaped progeria, right? So that's not always a good thing, but now here's these neutrophils that can make these different shapes, and that gives them access to areas so they can do their jobs. Like,
Yeah.
that's just such a cool, innovative idea, and it's like not straightforward, but it has the potential to really help, right?
I'm just very inspired when I hear your boss come up with ideas like that. It's like, oh wow, dang, that's where your brain is at. That's amazing.
It sounds like y'all are on the same page, which is really, really
Yeah.
cool of like coming up with these really fun ideas. And by fun, I mean like, it's like I'm smiling, getting excited, and planning 3 steps ahead too, and I'm not even in the field. Right. And so it's like, oh, that's like a cool idea to pursue.
Yeah.
So if we can, and Charlie, again, I know it's early days in the research. Please, if you ever don't want to tell us because you don't want to get scooped, we've talked about this here on the show before of like, there is competition here. Please, you never have to tell us any or all of the details. You could just be like, it's a work in progress. But out of curiosity, where do you start with this question? So we have a cell type and we have a hypothesis. How do you begin planning a model where you can give a tumor a mouse, a mouse a tumor? You can clear it so you can give the tumor again and make sure that when you're clearing it, it's actually gone, so you're not detecting previous iterations of it. How does one start that process from a technological perspective? And then what are the current molecular tools that you are finding to be kind of indispensable? Or maybe, I need single cell sequencing, but I need to adapt it 3 different ways before it's a big deal. Kind of like on that thought process.
Yeah, I heard a lot there. Definitely single cell sequencing will be down the road. I think if the audience is interested in that kind of stuff, I think one of the most important things for someone designing an experiment or a project or a thesis is start with a phenotype as fast as you can, and then you characterize the phenotype. Don't think like, oh, this is an interesting, unique thing that's happening. I wonder if it actually has any efficacy in the whole organism. That might be, although very cool to start, and you find all these mechanisms so fast. You might be a little bit disappointed when you hit the finish line, and only to realize the phenotype you've been characterizing is small. And so, where do we start with this kind of project? It's fun. It's kind of funny because I got my idea from reading a bunch of papers, so it's hard to say that the idea itself might be novel, but all of the tools have always been there, or at least in the past 5 years have always been available. So, I'm more of an architect of bringing these seemingly separate tools together and then looking for an observation that no one seems to be looking at. And so, Blend, I'm not entirely sure. I do have something, a novel tool.
Mm-hmm.
I have a novel idea. And in science, I guess we trade in novel ideas.
So it might be one where at least right now it's all technology that's available to you. You can make with like a
Yep.
unique combination, maybe that someone hasn't done before, right? To get you these
Sure.
answers. And then down the road, maybe you might have to come up with something new to get at maybe a spin-off question that has come from the original data set.
Yeah. And I don't know if I would say I invite scoopage in this situation, but I think that the finish line of an idea like this is such that you can call it scooping, and it might personally affect me, but to scoop, to call it, well, if they did it for me or for humanity, then there's a real good chance we hopefully have a better shot of fighting cancer. So one man's loss, mine would be humanity's win and the scooper. So I can't be too upset. There's more to life. There's more to life than work. And, I would just throw my hands up at that point and be like, all right, I'm going on vacation.
What a healthy mindset, by the way, just as an aside, right? Because it's like, we've talked about it here before. Some fields are very different than others, where it's like there is this hoarding of information because you're so nervous about someone
Oh,
taking the
terrible.
credit for it, right? Versus like the way you're saying it, like, guys, we're trying to save lives here at the end of the day, right?
Yeah. But don't scoop me, please.
Yes, guys, we want if we can get Charlie the Nobel Prize, we're good with this, right? But if we
Yeah.
know if we need to, let's talk to him first. Let's ask him.
Right, right, right.
So, what would be for you a result that would get you really excited by looking at this data set? Like, what is,
Yeah.
if I could tell you, Charlie, we're going to run an experiment tomorrow, and we're going to get whatever is the ideal result. What does that look like to you? What is a positive result here that is going to give you information to be able to act on long term to help people?
So, yeah, even right now with my idea that I'm, personally so sure of might be real, back in my mind, it's still telling me you might be wrong. You might be wrong. And there's a good chance I might be wrong. But the observation that I'm looking for to put all cylinders in and, pedal to the metal is every time I give that mouse a tumor, that tumor gets bigger, faster each time. If that's the case, that means that the immune system truly is losing the ability to contain the growth. And that observation alone would just blow the door wide open on the bandwidth that I'm have like juggling other projects. It would just be a singular focus because once that's been proven to me, I don't see how I could afford to spend my attention anywhere else. This is it. Yeah, that's the one thing.
Okay, so we have some spin-off questions of that I want to ask you some more about that because okay, so that makes total sense. Coffee asked a really important question here is when you're reinfecting with a new cancer in this mouse model, let's say, how is one doing that? Is it the same type of cancer? Is it the same location? Is it how I guess are you delivering it via injection? There's sometimes, right, we've talked about on the show humanized cancers where you take a tumor directly from a patient and put that in, right? So how do you start the cancer and then the follow-up infections, is that identical or different intentionally or not?
Yeah, that is an amazing question, and it seems like your chat's full of scientists. Uh,
Yeah. Can
yeah.
I just want to brag on this community. There are people in our chat who are like, I haven't taken a science class since high school and I'm in my 80s. And
Oh,
the questions that they ask, I could not have told you that these weren't colleagues who ran their own labs. We got a
yeah.
gentleman who's like, I've been a plumber for like the past 20 years and now I love GFP. And like it's to that level. Like there's really just this real love of and curiosity of the sciences, and that's who you're talking to today. So like yeah, the questions that come in are always like we dissect papers, and I'm just like that is a great question. I have no idea, and it's
Yeah.
like even we'll bring on the scientists who did the paper, and we'll get questions. They'll be like, yeah, I have no idea. That's a great question.
Yeah. Actually, Belinda, I have the memory of a goldfish. Can you repeat Coffee's question?
Yeah, so when you're reinfecting with that
Mm,
cancer, right?
yes.
Is it the same kind of cancer? So I guess number one, where does the tumor come from? Is it genetic on the part of the mouse? Are you transplanting a human tumor, etc.? Same kind of cancer, same location, or new kind of cancer, different location, or something in between?
Right. The cheatiest answer is just yes. All of these questions good and valid, and I love that. I mean, it's like Coffee's in my own brain looking at my own PowerPoint presentations. Those are the questions that we will be trying to address. And not only that, I'm also interested in looking at not only the same site of infection, but I mean, well, the site of implantation, but the sites that didn't get the cancer. You would assume that the whole mouse is changing system wide. And so I want to see all of it. Once I get this pilot data in, I will be Oh man, the whole mouse. There will be no tissue to go to waste. Everything will be as interesting as it was because no one's looked at this before. And so, you know, I'll be looking at sites of metastasis. I'll be looking at possible primary tumor locations, all sorts of things. And that is an amazing question that is literally in my head.
that's that's our community. And I think
That's
it's definitely one of those where maybe the foundational research might be like in the same location, the same type. But as soon as you can start mixing and matching, that gets closer to real life, right? Because the first tumor in our bodies, again, like you said, isn't necessarily the one that is discovered. So I might have one in my lungs that gets fought off by my system, but it's a
Mhm.
colorectal cancer that ends up being detected, right? And so then you can start doing that mix and match and see like how the immune system might be altered if it's localized to the same area constantly versus system wide in different places. Like it it's definitely one that I see even more the picture of you saying over a lifetime you yourself can't answer all these questions that we're throwing at you already.
Yeah, and I want to point out to the work that doctors have put in and statisticians is one of the things that gave me this idea was just a whole handful of papers that keep track of patients with cancer and search. So right now, the best way to cure cancer is to have it surgically removed, and
Mhm.
because once it's disseminated and you require things like chemo and radiation therapy, it's just not clear curable. It's too disseminated. So while it's still in one spot, if you can get it cut out, it's probably cured. And a lot of these studies have shown that, say, like in all sorts, of breast cancer being one, it's very easy to notice a breast cancer and then have it removed and be cured. The patient be cured. Same thing with melanoma and things that you see coming, like prostate. All of these cancers are so predictable that there are so many patients alive or people alive today that have been cured of it. Now, of those people today that other scientists and doctors are keeping track of, we're beginning to notice that they have 2 to 8 fold more chances of developing either the same cancer or any of the other cancers. And so the data in humans are already proving, or at least suggesting to be true, what I suspect in mice. The problem with the human data is, humans live such complex lives that how there's no control there. And how am I going to convince anyone of my hypothesis? I don't show something that's extremely controlled.
Right, just because of like diet, right? We know how many things. Like we had Helen on Monday to talk about how the gut microbiome
Yeah.
affects cancer progression differently, and that how it's affected by diet and location, and so all of it, like that comparison, right, in a human is not straightforward and not easy to do. So, like, right, you have to make that additional foundational step. Can you tell us why lung cancer in particular is something that you were interested in? And it seems also, it sounds from how where we're speaking, it's maybe a little bit harder and more difficult to predict than other kinds of cancers. Is that a fair assessment, or what is it about lung cancer?
I would love to be able to sell lung cancer to you, and I bet McGarry, Dr. Houghton would have way more to say about this than me. But I'll be completely honest, to you in the colony here. Um, when I was looking for, so I left my postdoc job at MD Anderson, to find another one, and there are reasons for that, and at that time, Trump had cut funding to NCI. He cut and put the entire scientific community on red alert in terms of, will labs get funding? And so a lot of labs, when I was looking, were not hiring.
Mhm.
So, I would love to say that lung cancer is my passion and some greater being directed me here. Maybe that did happen and I'm completely unaware of it. But in reality, I'm here because McGarry heard my crazy idea and said, let's do it. And so, but I can tell you about lung cancer. Lung cancer is, it's one of the most common cancers. It needs a cure more than, I'm not saying rare cancers don't need a cure, they do. But, if we're going to start, we start where people need it the most. And lung cancer, one of the ones, very aggressive, metastatic, you know, it's one of those organs, and the lung, in terms of fibrosis, and when the lung takes damage, it doesn't heal very well you're going to need transplants, and you have all sorts of other problems like, rejections and things like that. So, you know, I can, and I'm sure Dr. Ho can go on and on about the importance of lung cancer. I'm here because it's fated. Does that answer the
Yeah,
question?
so it's one of those where, it's the lab that you're in that you're working on, and it's not necessarily that this idea works the best in lung cancer models, right? Because the idea, like you were saying, is that in theory, right, you could be getting tumors from anywhere in the body contributing to this immune factor. It's just like you're just in the lab that's also specialized on this component, so you're able to look at it, and there's also clearly a need for it as well.
Right,
Yeah.
right.
So a question that we had from the chat too is how does one measure success in this model system? So like you mentioned in terms of a cure, right now it's just like popping the tumor out. And you want to
Right.
be able to have the mouse no longer be tumorigenic and then reinfect it and then again, no longer be tumorigenic. How does one do something like that? Like is it just going to be like grow a little tumor in there, pop it out, and do it again? Or like what is the process behind that and how does one measure when it's time to do that because of the unique nature of tumors, right? Like they can maybe start spread elsewhere in the body when there are different sizes, right? So
Mhm.
how do you know when it's time to pop it out?
Yeah, so I definitely want to pop it out before metastasis, because
Mhm.
once it goes, I don't, that doesn't model what I'm looking for, which is, if a tumor is resolved by your own immune systems, it didn't get very big, and so I only want to let the tumor grow for I don't know, like pea sized,
I
and
see.
then let it, and then end it, and then pea size again, and then end it, and so in terms of how to measure success, so measurements will be taken all the time. And like you had even brought up, the single cell stuff will be very important, especially in the immune populations. And once I identify the hopefully the phenotype in the neutrophils that have been altered by this process, we would strategically develop a way to counter that change. And then see if that counter change prevents the tumors from being larger and larger with each time. Ideally, what if we nerf whatever cancer is buffing, okay, we can hopefully just prevent it from growing in the first place. The idea is that if this change needs to happen for tumor to grow ever and overtake the immune system, if we prevent tumor from ever making that change, will people ever form tumors, or will form tumors just go away? So, there are some lofty ideas there. I'm definitely dreaming, and it's easy to dream in the beginning because the mountain of negative data hasn't beaten me down, but we'll see what happens.
So okay, so I guess that gets to the next point of what is that positive result again, right? So we talked about like, Charlie, you're going to get a result tomorrow. What is it going to be? And we talked about how increased I guess infection with a tumor, right, results in a further spread and a decreased immune activity. So I guess step 2 then is like you said, looking at the neutrophil populations in particular and seeing what are the genetic changes associated over time with that decrease, right? Assuming that the hypothesis pans out. Do you have, even before you've started, are there genes or gene groups or families that you're thinking might be affected if this were true? Like, is there a direction that you're already kind of like, it might be this zone? And that's not to say that you wouldn't be open to beating anything, but just based on what we know about the behavior of these cell types.
Yeah, again, totally. So, I mean, Glenn, are you a member of Dr. Houghton's lab? Because, yes, again, like I said before, I'm not some visionary who invented neutrophils' involvement in cancer. I'm merely stepping into what has been going on for decades. And so I would be surprised. It's definitely not a gene that no one's ever heard of.
Hmm.
Whatever the process is, I would even put money on someone's already characterized it. And who knows, maybe it's an extremely popular gene. So context means everything, I think, in science and in reality and all things. That finding whatever those genes, the 5 genes, no more than 5, Belinda, we went over this, no more than 5 genes that are important for this, it's a really good question. I'm curious to know and under what context it causes the phenotype that I pretend to be true. But I do not think that it is it the names of those genes will not be like, oh, I've never heard that, or I get the privilege of naming the gene myself after my late cat,
So we're
Rest in
assuming it's presumably a studied biological phenomena that is now altered because of maybe overstimulation. Is that a like? I guess that's
like
the other thing too. Is
insomnia.
why do
Yeah.
we think that these neutrophils are not as good as they are at the start? Like, what is it about having a tumor that would cause them to kind of go downhill like this? Is it just one where it's like the equivalent of going into a smoky bar, sit 5 minutes, and you no longer smell the smoke? Like, is that what's going on with our neutrophils? Or is that going to be like the we'll know once we sequence kind of question?
Oh, that's the boring way to respond. I can pretend. I can pretend to know at least. Well, neutrophils are already very well characterized in promoting how tumors grow, and like we said earlier, turning off T cell involvement and clearing that tumor. And so, the simplest way of this phenomenon happening would simply be there being more neutrophils everywhere. And so, I'm curious whether or not patients or mice just simply have higher levels of neutrophils with each instance of tumor of the body seeing a tumor. And so it's not actually unheard of in the field. And there is preliminary data to suggest that this is true. And so the real question is whether or not that upregulation of neutrophil presence we see can significantly impact tumor growth systemically. And there are some thoughts. Even if I fail, I'll be so sure that I'm right that I'm going to be figuring out about maybe I got the context wrong, and so maybe it's not each tumor implantation that promotes the next tumor, but maybe there's a threshold that must be reached, something like neuron action potentials. Maybe it's the first 10, and then once you breach whatever dam was holding back this phenotype, number 11 goes wild
Hmm.
out of nowhere like an action potential. It's something crazy. They I can see why the phrase mad scientist gets thrown around so much, because even when we fail, we have to find another context to prove ourselves true. And you can call it insanity or stubbornness, but without that kind of persistence, I don't see how medicine progresses. So I'll probably be some kooky. I could end up in a cardboard box under a bridge, but right now there are PIs out there who believe. So
I mean, that's I think the coolest kind of experiment, right? That regardless at the end of the day, whatever answer that you get is still going to be informative, right? Because like I think okay, if I was a funding body, you got me convinced, right? Because it seems like there's a lot of like there's clearly a lot of thought put into this, right? Like you're not just like off the cuff shooting at this. It's like you've taken us through this logic of a very elegantly designed experiment with a lot of potential to at least answer fundamental biology questions in the context of your immune system and possibly even in a disease state too, right? And so either way, whatever you're going to get is going to be cool, and eventually we'll just get you back on the show so we know what you ended up finding, right? But we have I think a very relevant question in terms of the end game of this, and there's actually a couple of questions off of that. So I'm just going to read out Glued with Gold's question is so the end goal. Is it eventually to develop a treatment that would repair or reinstate the immune system after cancer, or more looking for a way at the immune system to be able to detect cancer sooner, or both? So, again, Charlie, the question being, is this something after detecting a tumor that we want to kick back on the immune system, or is it something that we habitually want to keep activated so that we can detect cancer, or is it, again, both?
that is an awesome question. In fact, Murph has actually expanded on that. That's something that I would love to enter as my history as a biomarker early detection cancer biologist. Is if it is such that these small tumors growing around the body inflict larger changes on the immune system systemically, when that change that we identify happens, that of the outgrowth, if we could just sample a patient's blood and
Mhm.
observe those immune changes, it would imply that there is a large tumor growing. And so all patients would have to do is, or anybody would have to do is give blood once a year. And once, hopefully, if we observe those changes that have yet to be discovered, we can now justify an X-ray, an MRI, a more detailed pat down of the body to try to find that tumor that the immune system seems to be telling the doctor is present. So 100%, that's an amazing follow up to, wow, I mean, you guys in the colony have created next steps beyond my lifetime. And, that's why PI start labs, because they'll have a branching overall umbrella idea. Then that one idea turns into 100 ideas, and suddenly postdocs, grad students, technicians, research specialists are need to be recruited, to do all of that work. So yes, 100%. What Murph just said, yes.
And I guess a follow up to that is maybe this is outside of the field of what you're immediately trying to look for, but let's say that you find the genetic agents that are the causative factors of why there is this decline. And, in the mouse model and then maybe eventually in the humans, how does one go in and correct that? So, what would be the method to go in and fix it? Is it, do I cycle through your neutrophils and like put them back in you and they're adjusted, or how does one actually go about fixing something like that?
So feasibility is definitely a thing. What cannot be solved with a 24/7 medical attendance, with machines replacing your blood with something amazing, totally.
Yeah.
But what comes practical is we got to give them some magical pill that solves the problem, and they can only take it, once a day, twice a day at most. Because if people have to walk around with a staff that has constant infusions, it's just a terrible treatment approach. So the dream is it's an enzyme, because enzymes are the most targetable proteins that we can hit. What I would be less enthusiastic about trying to cure is if it's something like a transcription factor, something without a catalytic site, something that if you do one change, it affects 1,000 genes. So a transcription factor, if altered or muted or deleted, will change the function of so many downstream genes, not just the one that I'm interested in, that it's like, oh, dang it. So then we'll have to look for ways to treat downstream of that transcription factor, hopefully to collect as many of the, if it is the one transcription factor that is ultimately in charge of 1,000 genes, but only 100 of them are important in the phenotype that we care about, then now we got to find out a way to pick up as many of those 100 pieces as we can. Now, that's the worst case scenario, I think. So it's got to be something with a very specific activity, and I'm just hoping it's an enzyme.
Do you have a hope into what direction that you need to fix it? Meaning, is it something, would you rather find something that's overactivated in the disease state or underactivated, or does it not matter? And I guess could you comment too of like what would then be the difference in terms of how you go about fixing it in a patient or even in a mouse model?
That's a fun question. I want to think about that. I haven't thought about this. So if the target is overexpressed, which is causing the phenotype, then let's say we have an inhibitor to turn it down. So then if there's too many of that protein, they'll take a lot of the drug. A lot of the drug means a lot of side effects, cost of production. So that would kind of suck. So now let's go the other way. If it is a loss of something, how can we bring it back? If it's lost, that's a fun question, Belinda. I would say I would prefer to be a loss of function.
Okay. So, and just for the crew, that means that you would want it to be in the disease state going downward, that you would
Mhm.
have to target specifically a one kind of thing to push back up.
Yeah.
And presumably that's because of specificity, right? Is that the reasoning that it's not like the turning down a transcription factor that could be affecting 1,000 genes? It's that you just want to crank up one particular target back up.
Right, like specificity of dosing, because
Hmm.
I wonder what happens, it kind of like the problem with tumor and tumor heterogeneity and treating tumor sizes is if the tumor is this large and the problem protein is an overexpression, and you need 100 molecules of drug to cure or kill every tumor cell. Well, the tumor cells on the outside will see enough to get 100, but as you get deeper into the tumor, they will not receive the necessary 100 molecules of drug to be depleted. So if it's an overexpression, you're like, dang, I'm never going to get enough drug in there to do the job right. Whereas if it's something that is low but needs to turn on, It may only need something as low as one extra copy of what we're hoping to improve on to revive its function. And so I would, and I'm saying this with no experience in pharmacology or drug design. Let's say,
But that's not the point, right? Because like the cool part is like you're a researcher and I'm asking you all these hypotheticals and we're just coming up with experiments and like results that
oh,
how we interpret, right? And that's the beauty of this. That's going to be changing. Like if we bring you back on in 6 months, the answers might all be different because of what we found, but that doesn't invalidate any of
six
the
months
things that
to
we.
win. I'm not a race car. Have me back on in 2 years, and then I can tell you something new and interesting. But 6 months, wow.
The thing is, we usually don't get enough out of a person in like one of these interviews. It's usually like, oh, just come back in a few weeks. We'll just keep chatting. But no, I feel like that totally makes sense. And again, given how we've discussed how the system works, it would be the way to go. We have another really interesting question is, have you thought about using, for example, an autoimmune disorder model? So one where the immune system is already perturbed, and then using that as a means to kind of further validate any finding that you may have. Meaning that it's already not activated in the right way. If you put into an immune, deficient mouse or autoimmune activated mouse, the tumor model that you're proposing, could that help you narrow down your potential candidates or even validate it?
I think that's a very interesting question. That's one of those things that's down the line. I think definitely the thing about, maybe I need to touch up on my immune models. I'm not into immunodeficiency models. To my knowledge, we got the NOTSCID, we got the RAG, and the problem with me I'm a little apprehensive to jump in there, at least in the infantile stages of my research, because they have although they have 1 to 5 genes altered, so much is altered because of what those genes do that it's hard for the control. Like I'm trying to think, you wouldn't just change one thing. You've lost in one model all B cells and T cells, and another model just the T cells. So, and even when I say just the T cells do like 1,000 things. So I would like to keep it as controlled as possible. Although that's not to say even using a wild type mouse is not complicated. That's a good question. I never thought about that. What I can say about immunodeficiency is the observation is there as well. There's a crazy, crazy unfortunate observation in when patients who get an organ, they need to take immunosuppressants to promote that organ from taking, and it doesn't get rejected. Patients who are on those immunosuppressants when they receive a new organ are 2 to 5 times more likely to get a tumor during that time compared to a healthy person, which is just to say the moment you turn your immune system off, that immunosurveillance needs to be there all the time, or else tumors are very opportunistic. They're just waiting to grow. And so, that was one of the inspirational pieces of one of the inspirational observations that helped me formulate this hypothesis is exactly what the question kind of gets at. What can we glean from immunodeficiency situations? And that's I love the question.
Yeah, I think it's definitely one of those downstream target questions, like once you already have candidates, but it could be a unique way to, like, it's kind of just, again, you're expanding foundational immunological knowledge to other diseases now also outside of the cancer scope. But it could very well have something to one point look at. Charlie, I know it's a little bit past our hour. I want to be respectful of your time. How much longer may we have you? We got a couple more questions that have come in, but I want to be respectful.
Let's do it. Let's do the 2 questions. We'll do 2 questions, and then we can do the outro or whatever happens. I don't know.
Perfect. All right. So, guys, make sure if y'all have any last minute questions coming in, please let us know. Please feel free to throw it into the chat now. One from earlier, Charlie, was about the potential treatment in terms of both in the mouse model that you're going in to correct something, as well as in the person. Is this something that would be a potential one shot of taking care of, or after, let's say, this early detection, like let's say we can do a blood test or stool test to find that these genes have decreased and we need to start looking at you longer and re-up your immune system. Is that something that could just be a long-term thing? Because I'm almost imagining that even if I did crank it back up, the process of getting tumors over time is just going to continue, and eventually that same phenomena is going to, happen again and again and again. What would you say to that idea?
I would say your suspicions are 100% right. In fact, I would put money on everything what you just said to be exactly what happens, and what needs to happen is you got to come in for your refreshers. You're once a year. You're once every 5 years. And that's just the unfortunate state of our bodies. Is we're in once you passed like your 20s, you're in a constant state of decay. So we fix
Thanks.
your problem. Yeah, I know. I'm in my mid 30s. I feel the pain. I've been I tore my Achilles doing literally just waking up. I woke up with my Achilles torn. I didn't know that was an option. And when
Yeah.
people ask me, "Oh my God, how'd you tear your Achilles? Did you? Are you an NFL running back?" Blah blah blah. I'm like, no, no, no. I just simply woke up like a normal human, and my Achilles came torn. Welcome to your mid 30s. So, that's the thing of our bodies. That this treatment, and like I would argue, all treatments that will just have to be refreshed.
That's okay. So it's definitely one of those where it's going to be similar to the methodologies that are being dealt with tumor genesis and cancer today, where again, like if they find one within you, right, or like one example of it, then you're going to get retested over and over again and ensure that either A, that it's been cleared, it hasn't spread, etc. It's just going to be maybe part of the natural regime, if you will, to make sure the patient is healthy, which is
Yeah,
again really really. I mean, it's one of those where it really doesn't matter. Again, you're helping someone. It doesn't matter if it's not a one shot, right? Like maybe it'll eventually be a one shot, but it's cool to hear about. Again, it's an example of any part of your work, whatever you're discovering is gonna lead to something cool, right? And it's gonna be
yeah.
lead to something actionable in some way, and that's kind of the
And
best
someone
flavor.
smarter than me will make it better and more efficient. And so
Hmm.
that's just the way science is. It's I'm so fortunate to be. I should say we're also fortunate to be born. At the time we are now. I mean, could you imagine if we didn't have potable water, or we're running away from dinosaurs or something? But then you also got to ask the other question: what if I was born 1,000 years from now and we're in Star Trek? So we're not the best time. We're not the worst time. We're let's make that stuff.
It's exceptionally mediocre.
We're in an essentially mediocre time where I'm trying to think about it. I gotta go to PT instead of getting a new robot foot or something.
It's like from the chat. It's like, how'd you wake up with terrible back spasms? Was it hot
Uh,
yoga? No, I sneezed.
yeah,
Yeah.
exactly. Yeah.
So I guess our final question before we outro is, what are you most excited about in terms of either it being a piece of data that you're looking to collect, an experiment to run, or is it even something that's, actionable years from now? Like what is it that kind of is driving you forward on this beyond just the idea of like curing cancer? Like, on the more smaller day-to-day scale, like the idea of curing cancer, that can motivate anyone, but how do you keep going? Because you mentioned kind of as an aside, like the negative piles of data haven't really shot up yet. Even if that does, what is it that gets you excited, getting you out of bed and just continuing on with your research on just a day-to-day basis?
Mm, that's an amazing question because there are days where even in the 5 months that I've been with Dr. Houghton, that you know data has come back, or both either technically underwhelming, or actually underwhelming, even in the control situation. That the next day I wake up, you sigh a couple times and you're like, damn, I gotta go to work still. What keeps me going? I think it's the pressure from failing. You fail once, but if you give up, you fail forever. So that's a stick way of pushing yourself forward. I wish, and obviously the carrot is everything working, but what can I say? I think it's very important. So I just moved to Seattle, from Houston, but more just before that, New York, and where I got my PhD at Penn. And I think friendship and just doing fun things and unwinding, I guess, ultimately keeps me going, because moving to this new city and losing my support system, it really, the silence is deafening. And so I'm fortunate right now. I'm making some new friends. I just started playing Magic the Gathering. And again, I used to play in high school and then sold it all for me and my brother sold it all to make ends meet for prom. And then because I sold my collection, I never came back to it till now, that I'm in Seattle, and I'm making friends, and things are looking better. You got to have a strong support system to keep you going. No one can do this alone. It helps that you become friends with your coworkers. Cliche stuff like that, but Blint, that's all true. It's all true.
Yeah,
You
Helen
know,
had teased us with you getting back into Magic the Gathering, and that was her pitch. Like, you're gonna love Charlie. He's into Magic, and I was like,
I'm so into it.
I
I'm
was like,
so into it. My wife couldn't be more disappointed. She's like, "How much did you spend on Magic?" I'm like, she's like, "Well, I want to buy this bracelet." I'm like, "How much?" She's like, "Oh yeah, you should totally get it." She's like, "Wow." I'm like, because it's still not how much I spent on Magic.
see, if you really
Yeah.
wanted to troll her, you'll come home one day with just like a Magic hat. Be like, "This is the Magic I've been talking about." Just be like, what? I thought you were playing the card game. No, no,
Oh
I've
god.
been showing kids in the hospital magic tricks. That's what you've been getting at.
Yeah. I think she would like that a lot.
No, I think that's really, really important, and it just goes to show for the folks in the chat of how much it humanizes the researcher as well. That it's like you aren't like your background, right? It's not machines behind you. That's not you. You are a person. You also have the feelings of something failing. You have the feelings of something working. You have alone and hardship and friendship, and all these things are still coming into play. And it's just something that kind of news reports is kind of glossed over and lost. And so thank you in part, sharing your knowledge with us tonight, but also sharing that human aspect that's really important to maintain when talking to a researcher.
Yeah. It's do people see us differently? Is that a thing?
Oh, 100%.
Oh no,
Yeah,
why?
well, because at least the experience that I've had is people like big
Yeah.
scientists are in an ivory tower and they talk down. Like
Oh,
my mentor would tell me that science outreach is useless because people are too stupid to understand the science that we do, right? So it's very much this ivory tower mentality. It's not bringing in people to the fold. It's isolating, right? And that makes it very difficult then for anyone in the public to want to or even attempt to connect with us. And so as you're telling us, like, I have a hobby, I have, my significant other, I have these fears, I have these goals. It's like, it makes it relatable and it makes a world of a difference to put that in.
I can't apologize enough on behalf of those scientists who talk down on people. They're just awful people. Like it's a scientist independent. I think they just suck. Um, uh, yeah, I do want to. I do want to end if I can. I think it's if I guess some people do view scientists in a way that maybe and I don't want to be too presumptive, like, oh, they're smarter or more logical or whatever it may be. I just really wish, and I know I think there's a governor or two that have PhDs that are in the political sphere, and I just wish that scientists would do more than science. So Helen has given me this wonderful opportunity to work with you in your colony to spread the word of science and just talk about it, engage the community. And I just really wish more scientists did that, because what good is all of that brain power if you're just, like, I don't know, being a dick at your bench. It's pointless.
That's very well said,
This is
Charlie.
R-rated show, right, Belinda?
We're family friendly, but that's neither here nor there.
Oh no, R-rated.
We'll take it out in post. It's fine.
Oh gotcha.
No, but seriously, Charlie, I think that's very well said, and I really appreciate you again talking towards that, but also just honestly spending your time today with us, discussing your work. I know we threw a lot of questions at you. I also love the fact that you were like, "Man, these are some good questions," and it's been a lot of fun. So we really appreciate your time today. And if people wanted to reach out or support you in any way, is there any place that you would point them towards getting more information either about your research or just supporting you? Where would one go?
Well, definitely go to Helen. I think Helen's the was the leader of the philanthropy department at Fred Hutch
Yeah, she's
Or
one
if
of
she's
those
not the leader, she's like,
the
yeah,
integral members of making it all work.
it's,
Her, Jackie,
yeah,
Kirsten, Guppy are just an incredible team.
totally. And maybe by email. I don't know. I can type it down here. I have no idea how to use Twitch in this format. I sometimes watch VODs of game tournaments, but that's this is an interface of Twitch I've never seen before.
So
And
you weren't expecting a science show on
no,
Twitch.
yeah. And a huge shout out to my partner Joy. She had to take the bus home today because I had to do this show here at Lab, and so the sacrifice is appreciated.
We appreciate Joy for the joy
Yeah.
that you brought us. Look at that.
Oh,
See, there's joy everywhere.
she's never heard of that one.
I figured not. Yeah.
Like an overshare. Last name Ho. I've heard it all. Don't even try.
Listen, I'm Hungarian, so the number of times I heard, "Boy, you hungry?" living down in the South where growing up. Yep.
Oh,
Like, oh no, I've never heard. Nope, never heard that one before.
yeah, I know. That's I was an Asian ho in Tennessee, so I feel you, Bullen.
Yeah, you.
Middle school was savage. Yeah.
Oh God, yeah. But hey, but you know what? You came out on top. You're gonna be the one to cure cancer. Where are they?
Oh, where's Wood? Knocking. Okay. Yeah.
Thank you. Seriously, thank you again, Charlie. We really appreciate it. And would love to have you back anytime, whenever you're ready.
Years.
We got you. 2 years. All right. We'll be reaching back out. Thank you, Charlie. Have a great evening.
Take care.
This is for everyone, all backgrounds, all ages, all genders, and all walks of life deserve an education. Come be one of us.